I would like to invite you on a journey. Let's travel together to the future. Imagine it is 2050. You are admitted to hospital for a hip replacement. This is a surgery which we consider today as standard and safe. But before the operation, your doctor tells you that the biggest risk is not the surgery itself, but if you would develop a bacterial infection, there might not be an effective antibiotic. In 2021, bacterial infections have caused more than 1 million deaths, and estimations predict that this number is going to rise. Antibiotics are so important and have been so effective that we have forgotten how life was like without them. So my name is Jeannine Hess. I'm a group leader at the CRIC, at the Biological Inorganic Chemistry Lab. I'm a senior lecturer in the chemistry department at King's College London, and it is my great pleasure to welcome you today to my crash course in next generation antibiotics. And this will be in the context of how we use inorganic chemistry to come up with new solutions for the future. My research group is working tirelessly to find new innovations that your potential hip replacement won't become a problem. So let's get us started by understanding why we need antibiotics, why they are so important, and why they are potentially different to other drugs we're more familiar at. So bacteria are all around us. They are everywhere, and they play really important roles in maintaining the environment we live in. So we have millions and millions of microorganisms on our body, on our skin, on the outside, but also on the inside, in our mouth, in our intestines. And most of them don't do us any harm, but some of them can cause infection and disease. And to actually keep those bacteria under control, those troublemakers, we need treatments and drugs. And this is why we need antibiotics. So an antibiotic can kill a bacteria, or it can stop the growth of a bacteria. And they are really essential to keep our healthcare setting at work and to actually control your health and the public health. But antibiotics have to do something incredibly challenging. So they should only actually kill bacteria, while they should leave our own eukaryotic cell unaffected. So you can think of this as if an antibiotic is the key to your house. You only want to get into your own house. You don't want to get into your neighbor's house. So how do an antibiotic select for a bacteria over an eukaryotic cell? So let's look at this illustration. And at first glance, we could say bacteria are much simpler than our eukaryotic cells. You can see that the bacteria doesn't have a nucleus. They don't have any membrane-bound organelles, and they have circular DNA. While our own cells, we have our DNA in the nucleus, and we have other organelles such as mitochondria and Golgi in order for our cells to keep reactions defined, to have them at a certain location. But these differences and this so-called simplicity maybe of a bacteria makes it really hard for a compound and drug to be effective. And this is why antibiotics usually just have a few targets, a few ways in which they can kill bacteria. So some of the antibiotics, they basically interfere or disrupt the membrane. It's like they are protective coat and barrier to keep them intact. Other antibiotics interfere or mess up the protein synthesis. So they take advantage of key elements that the bacteria need for their survivals. And other antibiotics mess up DNA replication. So all in all, we have much fewer targets available. But actually, even if you have the perfect target for an antibiotic, this is only half the battle, because actually for an antibiotic to be effective, you need to think it needs to get into the bacteria, it needs to stay inside, it needs to accumulate, and then it needs to reach its target. And bacteria are really smart. They find ways in order to escape antibiotics. And one way is, I told you about this protective wall, this membrane. So some bacteria, like gram-negatives, they have an extra wall, but also they can change that wall in response to an antibiotic. Think of it, if you came here and it was raining, you would have probably put on your raincoat, right? So this is how bacteria can react to an antibiotic so that they can escape that treatment. Another way how they can react is, even if the antibiotic enters, they produce pumps, and these pumps can kick out the antibiotic again. And all of these, or some of these processes I described to you now, is what we know and what then basically leads to antibiotic resistance. This is also covered under the broader term of antimicrobial resistance, where microbes, such as bacteria and fungi, can escape the treatment of an antimicrobial. So if we look at antibiotic resistance, you have a certain set of bacteria, and they are treated with an antibiotic, but some of them can survive. So the surviving ones will stay, they will grow, and they will take over. And then they can pass on their resistance to other bacteria, and thereby accelerating the problem. So I have shown you this, or we discussed that a couple of ways how bacteria can escape an antibiotic: changing the protective wall, producing pumps to kick out the antibiotics. But there are many, many more ways how they can react to antibiotics. And just to mention a few, for example, bacteria can produce enzymes, little machines that can break down antibiotics. So I refer to them in my talk as scissor proteins. They can come and chop the antibiotic so it is no longer active. And there are many more ways. So, for example, they can change the target so the bacteria is no longer can no longer be killed. And all of this together contributes to AMR. And the global burden of bacterial AMR was over 1 million, which was directly attributed to bacterial infection. So every year, there are actually 5 million deaths, which are directly associated to antibiotic resistance. And then this translates to 1 death every 6 seconds. And estimations predict that this number is going to rise in the future. So by 2050, we will actually see, or potentially will see, more than 10 million deaths per year. And this will then, or could overtake cancer. So I understand that these numbers might be overwhelming and might come as a surprise to you. But let's be clear here. Antibiotic resistance is not a sudden crisis. This did not happen overnight. So in order to understand why we are here and how we got here, we need to go back and think of how have we approached, for now, antibiotic drug discovery. So I have to go back quite some time. I think that's really interesting. So let's look how ancient civilization have dealt with bacterial infection. And there is really good historical evidence that they have used natural remedies and treatments. And the best well-known example or best documented example is by John Parkinson, who actually used moldy bread for topical wound infection. And he has reported this discovery and observation in this lovely book, Theatrum Botanicum, in 1640. And from there, it took some time until Paul Ehrlich really studied how we can use compounds synthesized in the lab to then treat infectious disease. So how did he come about this idea? So he was first interested in studying synthesized compounds and see how they would react with different type of cells or tissue. And these compounds were actually dyes, so they could color the tissue. So from this observation, he realized, hold on, some compounds react differently with a certain tissue. So he understood that there is some selectivity depending on the compound. And this is when he actually thought, could we now use compounds synthesized in the lab to also treat infectious diseases? And this is how he then basically discovered the first synthetic compound to treat syphilis. This is a compound similar to the one here on the top right, which is an arsenic-based compound, which was later sold under the trade name Salvarsan. And much of this discovery from Paul Ehrlich was then actually superseded by, you may be aware, the serendipitous discovery by Sir Alexander Fleming of penicillin. So Fleming was a professor here at the St. Mary Hospital in London, where he studied Staphylococcus infections. And after returning to his lab, he actually saw that in one of his dishes, so what you see here on the right-hand side, that is a petri dish with colonies growing. But in this dish, there was also a green-blue mold growing. But next to that mold, there were no Staphylococcus colony growing. So at the time, he actually said, this mold has produced a juicy substance that has killed my Staphylococcus. And this juicy substance is basically what we know now as penicillin. And this has, of course, saved millions and millions of lives. And this made him then also say that he did not invent penicillin, nature did it, and he just discovered it by accident. But this idea of using compounds produced by nature to kill bacteria was then actually taken on by Selman Waxman, who really utilized this idea to really use those naturally produced compounds to kill bacteria. And he went even that far that he said an antimicrobial is a compound which is only produced by a microbe to then kill another microbe. And this idea then really kickstarted the discovery of many antibiotics and led to this golden age of antibiotic drug discovery between the 1940s and 1960s. So here on the graph, you can see that many, many antibiotic classes have been discovered in that time. And now that seems funny, but in 1960, people actually felt, we have solved the problem of bacterial infections. We have enough antibiotics. This won't be a problem. And this then also led that much fewer antibiotics were discovered. One could argue the low-hanging fruits have already been picked, and it was much harder to find new treatments. So I haven't shown you something. This is only when an antibiotic class was discovered with these black dots. I haven't shown you that soon after an antibiotic is used for killing bacteria, it will also actually increase the chance of spreading resistance. So as soon as you use an antibiotic, you will likely get resistance. Resistance is really hard to avoid. And this is why antibiotic drug discovery, although so important, is extremely challenging. So in the last 25 years, in this review paper, they have summarized the number of antibiotics that have been introduced to the market so that they can be used in the clinics. So on the y-axis, you can see the number, up to 7 is the maximum. And then you can see each year how many antibiotics reach the market. So I did the maths for you and summed it all up. So over 25 years, only 51 antibiotics reached the market. This is a very small number. And I want to show you and put this a bit in perspective, not that you just have to believe me. If we look at the FDA report from last year, so here you can see different therapeutic areas and number of drugs approved. And you can see oncology stands out. So let's focus on this. From this, we know that only in oncology itself, 16 anti-cancer drugs have been approved just last year. And that's, of course, much fewer than antibiotics. And what is actually hidden in this graph is that many of these 51 antibiotics approved are modification of old drugs we know from which were discovered, especially in this time between the 1940s and 60s. So they are just slightly modified to change their activity. And I would like to illustrate to you what I mean with modified so that you have actually a visual context to this. So on the left-hand side, you see an example of ciprofloxacin. This is a fluoroquinolone antibiotic approved by the FDA in 1987. I have highlighted the core scaffold of this fluoroquinolone in pink. And you will see that 30 years later, a similar compound was approved by the FDA. We can just say it has some additional functional groups, some additions to the original ciprofloxacins, which are, of course, important. This new drug will expand the lifetime of fluoroquinolones and will have some different ways in killing bacteria. But ultimately, this is not a new structure. Bacteria have seen it before, so they know how to react to it. And this is what my group is now working on. We want to come up with totally new structures that bacteria have not seen before because they're not from nature. So now I would like to invite you to my last part where I illustrate you a current research project in my group where we use metal complexes. This is why metals to the rescue. So let's go back to the basics. I hope you do remember a periodic table from your studies. So in this periodic table, we have different elements. And so far, I have shown you different antibiotics. These were all organic antibiotics. So what do I mean with organic? These organic compounds are built from a number of building blocks like carbon, nitrogen, oxygen, hydrogen. You can think of it as if it would be your Lego set. You have a certain number of different colors you can use to build your antibiotic. And in my group, we actually want to extend the Lego set because we are now using this large proportion of atoms, which we know as transition metals, so we can form new structures. And metals play a really important role in nature and in our human body. We have it. You have it in your blood. We have iron, we have zinc, we have copper. So we are familiar to this. But what it means that if we use those metal atoms, we can actually build more fancy structures, which we refer to as having different 3D geometries. So with these different geometries, we have new scaffolds, and potentially we find new ways in how these compounds can kill bacteria, and they cannot easily escape those treatments. So as chemists, I do apologize. I like to categorize compounds. And let me talk you through this, because I think it's quite abstract if I tell you 3D geometries. So in order to illustrate what I mean by different 3D geometries, we need to look at this plot. And in this plot or graph, we can say how shape diverse a compound is. So on the really left, we have the rod-shaped compounds. You can think of them like as being a pencil shape, or a compound can be more like a disc, like a sheet of paper, or it can be sphere-shaped, like a 3D ball. And I hope you agree with me that the metal complexes we have in my lab, they are very diverse. They are in each and every corner of this diagram. And if we now go back to this organic compound I showed you, they are different. They are more like a pencil or a sheet of paper. And it's very difficult for them to be ball-shaped. So this is something really unique about metal complexes, that we can form these ball-shaped structures. So metal complexes are already used in the clinic. This is nothing new. And I would just like to give you some examples here. So the best or most well-known example is cisplatin. This is an anti-cancer agent used in many cases. There is also auranofin. You can see it is a gold compound highlighted in nicely yellow. The gold part, the gold atom, and is used for the treatment of rheumatoid arthritis. And then we have silverdane, which is a silver compound used for wound infection after 2nd or 3rd degree burns. And there are also other complexes here on the bottom, which are actually in clinical trials. So it means these compounds are now tested to be potential therapeutics in the future. So there is increasing interest of using metal complexes as therapeutics in the future. So I've showed you that metal complexes have really interesting 3D geometries, but they can actually do much more, which we are really excited about. They can also react in biological systems differently. They can lose some of their parts and then bind to DNA, so they stop replication. And they can also produce toxic species that can actually kill bacteria. Or as chemists, we like to use them as catalysts. So they're little machines to transfer your substrate into a product more efficiently. And all of this together, we use in my group to design new compounds to kill bacteria. But today, I would like to describe you in this project where we use another property of our metal complex. They can react with light. And these are complex processes, which we summarize on this diagram here on the left. And I mean, it's a detail. We don't need to understand this diagram. The basic message is that if we have a metal complex, we shine light, the metal complex gets to an excited state. It's similar to us, right? If we go sunbathing, we get excited about it. So just think of like being in that excited state. The metal complex can react with oxygen, which is all around in our body as well. And then it can form another form of oxygen, which is toxic and can kill the bacteria. So we used this idea to now design a new metal complex to kill our bacteria. But hold on, I told you selectivity is really important. And this is why we came up with some design here. So let me talk you through the next diagram. So here we have what we called a protein of interest. Just think it is a very important protein in a bacteria. So if we want to bring our metal complex to this protein of interest, we need something that would direct our metal complex to it. So I refer to this as a GPS tracker from now on. So we have attached a GPS tracker to our metal complex. So the GPS tracker brings our metal complex to our protein of interest. And then we can shine light. We can also decide where and when we want to shine light. And only where we shine light, the metal complex will produce this toxic species. And because of our GPS tracker, this is close to our protein of interest. These toxic forms will damage our protein and break it up. It will release our metal complex so it can find another protein of interest. And then this is what we know as a catalytic cycle. This can go on and on. So what was our protein of interest? I have told you that bacteria, as a response to antibiotic, they can produce these proteins, these machines that act like scissors. They can cleave and break antibiotics. And the protein we studied was NDM1, new daily metallo-beta-lactamase, which can cleave this beta-lactam antibiotic on the left. So on the left, the beta-lactam is active. In the presence of our scissor protein, you see, it gets cut open and it is no longer active. So this was our idea. We want to stop this scissor protein from working. So now, how did we find our GPS tracker? So we went to the literature and we saw that there are a number of compounds reported that are very specific for this scissor protein. So this is the compound here on the left-hand side, N1, a dipicolinic acid. So we attached our GPS tracker to our metal complex, so we end up with this compound in the middle, Ru1. And then we like controls. We need to make sure that the GPS tracker does its job. So we made a second metal complex which had no GPS tracker. So with this, we first had to test if these compounds still react with light. We need to know if they produce this toxic form. So just to really sum up this, we did a couple of experiments to understand how long do these compounds stay in this excited, happy state where they can react with oxygen. And we found that they stayed really long in that state, up to 480 nanoseconds. And we did some other experiments really to show what form of other oxygen species we form to understand that this could really damage the protein. But we were all set. We know our metal complexes can react with light and they can form this toxic species. So the next thing was we had to get our scissor protein. So we had our scissor protein, NDM1, and we did a couple of tests in basically where we studied some color changes. If the scissor protein is active in our test, we would go from a yellow to an orange color. But if we stopped the activity of the scissor protein, we only observed yellow colors. So we did a couple of, we call that titration. So we used different concentrations of our metal complex. First, without light, we saw the compound binds. It can stop the scissor protein from working. But when we now shine light, we see 100-fold improvement in the ability of the compound to destroy the protein. Next, we just tested the metal complex itself. Without light, the complex does not find the protein. Nothing is happening. But if we shine light, because the compound and the protein are together in one flask, they can still damage the protein. But this gives us another idea, because we then can compare the effect of our GPS tracker. And we could show that the GPS tracker is important because it gives us a 25-fold selectivity for destroying the protein. So next, we wanted to see what happens if we have our scissor protein and another protein present. So here on the top left, you have our GPS-tracked metal complex, our scissor protein, and another protein at different concentrations. If we don't shine light, nothing is happening to either of the proteins, and we also get like an intact protein from other experimental techniques. But when we now shine light, we could show that we selectively damage only the scissor protein and we don't affect the other protein. This was a really great result. So we wanted to go a step further. But for this, I told you, having a target is only half the battle. We had to understand, would our metal complexes even reach that target? And for this, we had to use a microscope to actually see what is happening inside bacteria. And something really exciting about metal complexes is that when you shine light on them, they glow. So we can directly see if they are inside or not. So here you can see our GPS-tracked metal complex inside bacteria glowing red. And we also used a couple of other techniques where we could show that all of the compounds we have studied entered bacteria. This was really good news, because now we could go only from our scissor protein to actual bacteria that would produce our scissor protein. So we did the same experiment, the same tests where we go when we study this color change from yellow to orange. So we can even stop the scissor protein from working inside bacteria. You can see here, without light, our GPS-tracked metal complex can stop the protein from working. And when we shine light, we see a 30-fold improvement. And here, very excitingly, the metal complex, which has no GPS tracker, and we shine light, has no effect on our scissor protein because it's not reaching it in the biological context. So we were all set for the next few final experiments to see if this could also save some antibiotics from stop being broken down. And for this, we had first to test if our compounds are antibiotics or not. So we had different bacterial strains, an E. coli empty, which means it's just an E. coli strain without the scissor protein. And then we have E. coli NDM1, an E. coli strain with the scissor protein. And basically, the higher the number you see in this table means that the compound has no effect on the bacteria. So you will understand now that our compounds are not antibiotics. They don't kill these strains or they don't stop the growth of them. And you can directly see at the end of the table, we have our control, meropenem, which is a beta-lactam antibiotic, which can be cleaved by the scissor protein. So you see in the empty strain where there is no scissor protein, the antibiotic, the beta-lactam, is active, so the number is low or small. And in the scissor protein strain, the number is high, so it is not active. So what we now did is we compared the combination of our complexes together with meropenem, the antibiotic, to test if we can save the antibiotic so it is no longer broken down. And indeed, just to show you these results in a table, which is a bit easier to digest, you can see that really excitingly, our metal complex, together with light, could stop the beta-lactam from being broken down by the scissor protein in bacteria. So with this, I would like to sum up the research project I have shown you from my lab. We have addressed an important resistance protein in E. coli and have used GPS-tracked metal complexes, new scaffolds that nature has not seen before. And we could show that this is a selective approach, that we can selectively damage our scissor protein. These compounds are internalized into bacteria, and we can save an antibiotic from being broken down. And I'm almost out of time, but I would like to finish with a couple of more thoughts and concluding remarks, because you might still wonder by now, but Jeannine, why is it so difficult to get an antibiotic to the market? And of course, I won't answer this question fully in 5 minutes, but let's just think about this complex problem in simple ways. So if an antibiotic reaches the market, it just will not create enough revenues, because even if we would discover an antibiotic today, which would be great, good antibiotic stewardship by doctors and nurses means that the antibiotic will not see a patient. You want to keep this antibiotic shelved to only use it when you absolutely need it, because otherwise we know that we will trigger the development of resistance. So this is why people like to refer to antibiotics as fire extinguishers. We have them, but we don't want to use them, right? They are there as a rescue. So this means that if we develop a new antibiotic, but it is not used, the production volume of this drug is really low. But here's the catch. If you actually want to develop an antibiotic, the research and development costs are as high as for an anti-cancer drug. And also, so are also the risks. So this is why the public sector and pharmaceutical companies have pulled out of antibiotic drug discovery, because the costs are just so high and you will never create enough revenue on the drug. So this is why we now see and need more public interventions to really fix this damaged pipeline. And just really briefly, there are different incentives which are summarized broadly as push and pull incentives. So the push incentives are government bodies or charities that provide funding for the upfront costs that people now invest again in antibiotic drug discovery to actually mitigate the risks which is associated with developing a new antibiotic. And then once you reach this critical point of an antibiotic is approved, there are these pull incentives so that the funds are still available later on, even if the drug is not used. So this is, for example, the subscription model of antibiotics. And of course, I would really like to finish with this thought that antimicrobial resistance is complex and it is a global problem. And AMR is present in all sectors and reservoirs, and transmissions are happening between human, the environment, the agricultural, and the clinical settings. And we all take part in this. So I would really like to urge you to only take antibiotics if you have a bacterial infection. We all take part in saving our antibiotics. And for me, of course, the most important slide is to thank my group, it is really an absolute pleasure to work with. They are there in the back. I'm very privileged to be working with such dedicated people who come into the lab and trying to find new and also unconventional solutions to the problem. So I'm really grateful to all my group members, former group members, the CRIC STPs, the NMR facilities, trick operations, and of course the funders. This research is not possible without substantial funding. And thank you for your attention.